So welcome everybody. Good morning, even good evening or good afternoon. So it is a great pleasure to have you around again for this new cerebral blood flow seminar series. I'm Patrice Brassard and I'm co-organizing with Caroline Rickards this series. So for those who are new to this series, I'll review a few rules of engagement and then I will present the chair and the speakers for today. So keep your microphone on mute and the video off throughout the session. There will be a time for question at the end of each of the presentation. So you can either use the raise hand feature or just send or write the questions in the chat section, and the chair will ask the question for you. The session, as usual, will be recorded and posted on the CARNet website within the week. And speaking of CARNet, if you are interested in contributing to the network, just send us a CV to Caroline Rickards. And for the moment, there is no membership fee. And we are looking forward to have the new members. So without any further ado, today we are really excited to have Dr. Jana Kainerstorfer chairing a session about methods related to autoregulation, and the title of her. So she's an associate professor of biomedical engineering at the Carnegie Mellon University, and today she will talk about near-infrared optical methods for assessing autoregulation, intracranial pressure, and neurovascular coupling changes. Dr. Kainerstorfer, it's to you.
Thank you very much. Let me make sure I actually share the screen. All right. You should be seeing the right one, right? Yes, that should be okay. Gotcha. All right. Sounds good. Thanks a lot for the introduction. Thanks for letting me organize this session. And I'm really happy to be here with three of my colleagues in the biomedical optics community. So I thought I would structure the session around individual research topics that are all related to cerebral autoregulation, but really the theme is how do we use optical methods, in particular near-infrared spectroscopy and derivatives thereof, to say something about cerebral autoregulation. So I really don't consider myself a keynote speaker here. It's more a joint session between four experts in biomedical optics. So really the title of my talk is it's very long and somewhat non-descriptive in a way, because I do want to talk about the potentials of near-infrared spectroscopy that some of you may not be familiar with, and just kind of take the opportunity and show you that there are additional instruments out there that may not be commercially available at times, but that there are many research groups that use those, and that there is additional potential for research, especially in the field of blood flow measurements and autoregulation. But really, my lab at Carnegie Mellon is located in biomedical engineering, but I'm associated with electrical and computer engineering as well as the Neuroscience Institute, and pretty much that combination tells you about my research. So we do a whole lot of instrument development and signal processing and such. And I do want to talk about intracranial pressure and influences thereof on autoregulation, as well as neurovascular coupling. But I thought I'd start out with just near background a little bit more. I think most of you should know that really those optical methods that are thrown around and are used, they're all based on the idea that we can use near-infrared light to say something about hemoglobin, specifically about oxy and deoxyhemoglobin. And if we use two colors of light, we can differentiate between the two species of hemoglobin in tissue. And that has been around for decades. That's called pulse oximetry. It's heavily used in hospitals. And typically the near sensors you might see in the market look something like that in one way or the other. You place optical sources that are really not any better than a laser pointer at times on the head, and either deliver it with fibers or directly an LED on the head or whatnot. And I keep forgetting the laser pointer function in Zoom. And due to scattering of light, we can actually penetrate the brain. So light goes through the skin, through the bone, interacts with the brain. Some of it comes back out again, and that's something we can measure. And companies are really building wearable sensors nowadays, or high resolution or multiple fibers on the head. But the general idea is kind of shown here, that we're sensitive to a blob inside the brain, and specifically then measuring hemodynamic changes in the microvasculature directly underneath or replace the optical fibers. And so to just give you an idea of typically what people are doing, if you're not familiar with NIRS, you can place sensors, let's say somewhere in the motor cortex region, ask the subject to tap their fingers, a typical functional task. And what you measure is intensity changes of that light that comes back out again. And whenever the subject is actively doing something, you see intensity fluctuations that correspond to that particular task, which can be converted into changes of oxygenated and deoxygenated hemoglobin, and we can do an average of this. And then whenever functional activation occurs, there's an increase in oxygenated hemoglobin and a decrease in deoxygenated hemoglobin. It's all relative changes relative to some baseline, and so on. So most of the people in the audience who have used NIRS will use an instrument that probably is really just giving you relative changes. If it gives you anything else, let's talk, because there is a whole lot of calibration behind that may not necessarily always be valid. It depends on the application. And so I thought I'd just dive a little bit into the differences between different NIRS sensors out there. And most of the commercial instruments that are available for user base are based on what is called a continuous wave instrument. That really just means you send light in, you measure whatever light comes back out again through the tissue, and that attenuation, that reduction in light can be converted to oxy and deoxyhemoglobin changes, but it's really not quantitative. If you want to know how much blood is in that particular region, which would be important, for instance, during stroke or during traumatic brain injury or any others. If you want to know how much blood in comparison to some other region, you may want to use some other instruments that can be more quantitative. And so there are quantitative instruments out there that are called either time domain or frequency domain. And the idea is really that instead of just looking at how much light do you get back out, you can quantify what is called a temporal point spread function, which really says if we send in a pulse of light and we measure how much comes back out again, we ask not only how much, but when exactly does it come back out again, so we can build up what is called a temporal point spread function. And the math here is really not that important for this talk. And by the way, I took this figure from a really amazing review article that just came out recently in 2019, if you're interested. But the idea is really that other instruments other than continuous wave, you might actually be able to differentiate whatever is happening in the skin based on looking at just early arrival photons versus much deeper layers, so you can do some 3D reconstruction. But really more importantly, the continuous wave instruments that are mostly used will give you temporal changes of oxy and deoxyhemoglobin, which is amazing and very powerful for looking at, for instance, functional activation or just changes over time. If you want to be more quantitative in terms of, for instance, knowing what is the tissue saturation, the tissue oxygen saturation of hemoglobin, how much oxy and deoxyhemoglobin is really in that particular region, or combined with blood flow, calculating the cerebral metabolic rate of oxygen, you really need some of those more powerful instruments, and that are some of them commercially available, but mostly in research settings. And so my lab, for instance, uses mostly a frequency domain instrument, which is commercially available. Yeah, I just want to point out that there's more that can be done with NIRS in principle. Also, what is often maybe not necessarily noticed, but the idea is if you can put many optical fibers on someone's head, they're actually a group like Adam Egebert's group at WashU, who's doing 3D reconstruction, have shown that you can reach, in principle, you can reach spatial resolutions of millimeters or like a few centimeters that reach fMRI quality for 3D reconstruction. And my own lab has recently shown that if we do multimodal imaging combined with electroencephalography, we can actually do source localization in EEG with using the optical NIRS reconstruction to come to really high resolution reconstruction of where neuronal activation has occurred. So just keeping that in mind, that NIRS can do way more than probably some of you may know, and I'm more than happy always to consider additional collaborations and see where we can take technologies that you may not have thought about. But one other method that is less known maybe in this community is let's call it a derivative of NIRS, which is called diffuse correlation spectroscopy. And what that does is very similar in its principle, as in you have a laser, you shine it onto tissue, it goes through the skin, through the bone, interacts with the brain, comes back out again, and we measure it. But instead of telling us anything about hemoglobin concentration per se, what we can measure is microvascular blood flow. And that's where really autoregulation also comes into play. So the instruments typically don't look any different. It's a box with lasers inside, optical fibers on the head. And really, I assume the other people in this session, probably Wes and Turgut, might talk more about DCS, but the general idea is that instead of just looking at intensity, well, laser intensities, we're looking at what's called a temporal intensity autocorrelation function, and the faster that laser autocorrelation function decays, the faster the blood flow. And the idea is we get a parameter that is really related to that local change in cerebral blood flow, so localized based on the optical probe. So in comparison to transcranial Doppler, for instance, you could really build up an instrument that gives you spatial variability in cerebral blood flow. But then also some of the signals that we do get out is like, again, this change in blood flow is a function of time. And so this is an individual heartbeat, cardiac pulsation in the brain measured on a healthy volunteer and on the forehead, just to give you an idea of what really raw, more or less unprocessed data can look like. So with that, I've spent about 10 minutes of my time just telling you that NIRS and DCS are optical methods that have a lot of potential. There are probably more applications out there and opportunities out there that you may not even think about. So while I talk about my own research, maybe think about how you would want to use any of those optical methods, and I'm more than happy to talk about that. But my lab is really focused on trying to understand how intracranial pressure and changes in intracranial pressure have an influence on cerebral autoregulation, as well as neurovascular coupling. And so intracranial pressure really dominated by three compartments: cerebrospinal fluid, blood, and brain tissue. If any of those compartments increase in volume, we have changes in intracranial pressure, which increases in ICP have been associated with worse neurological outcome. So it's an important parameter. And so typically the way I like to think about it is that we do know just the very basic description of autoregulation in terms of Lassen's curve. if perfusion pressure goes outside any of the autoregulatory range, so where perfusion pressure is really, we use the definition of blood pressure minus intracranial pressure. If perfusion pressure goes outside any of the autoregulatory range, we no longer have blood flow, stable blood flow. And coming more from the clinical perspective and the pressure reactivity index and optimum perfusion pressure ideas, people have shown that optimizing perfusion pressure in the field of traumatic brain injury, optimizing perfusion pressure, which I think goes hand in hand with saying maintaining cerebral autoregulation and blood flow regulation, has been shown to optimize neurological function and neurological outcome in patients with traumatic brain injury. And so where my lab comes in is saying, all right, there should be a link between, or we know there's a link between autoregulation and cerebral perfusion and perfusion pressure. And blood flow itself goes hand in hand with neurological function, and so therefore neurovascular coupling. So we're saying there should be a link between cerebral autoregulation and neurovascular coupling, which some of you have looked at already, and I'm just going to go into more detail there. But then the other way around, we're saying, well, can we use neurovascular coupling as an indicator of cerebral autoregulatory health? And so while the description of autoregulation in its basic form with the Larsen's curve is very nice, Larsen's curve has often been described with the x-axis being blood pressure instead of cerebral perfusion pressure, with the assumption that intracranial pressure stays constant. Now, that is definitely not always given, especially in cases such as hydrocephalus and traumatic brain injury and potentially stroke and definitely SAH and so on. And the issue here is that mostly in those patients, if there is not an intracranial pressure sensor available, you don't have access to perfusion pressure. So since ICP is an invasive, the clinical gold standard for ICP measurements is invasive, put a pressure probe inside the brain. We thought, well, can we develop a non-invasive alternative with actually the goal of saying something about perfusion pressure and therefore autoregulation. And then we also measure neurovascular coupling, and I'll talk about that for a few minutes. So really, from my lab's perspective, the overarching goal is to say, how does perfusion pressure and ICP change neurovascular coupling? How's neurovascular coupling? What is the relationship between neurovascular coupling and cerebral autoregulation and blood flow? And all of this combined, can we develop biomarkers that are based on neurovascular coupling, really, to say something about cerebral autoregulation? So now I do want to talk about neurovascular coupling for a second, because mostly when someone is using NIRS or transcranial Doppler or fMRI studies, what we measure is hemodynamic changes. We measure the vascular response to a neuronal change. And most of the times, in healthy population, we can assume that this vascular response corresponds to a neuronal activation. The coupling itself, though, is actually described by what is called a hemodynamic response function. So the idea is that we have neuronal changes that will trigger hemodynamic changes, and they're coupled. And that coupling is something I want to talk about. So just in terms of neurons, the coupling is described by what is a convolution product with an impulse response function, which is called the hemodynamic response function. So neuronal changes we can measure, for instance, shown here with the visual evoked potential. So that's electroencephalography, electrodes put on a visual cortex. The hemodynamic response we can measure with NIRS, as I said. So that kind of looks like something like this. And really what we have been saying is this coupling, the description, the function itself that links neurons and the vasculature, the hemodynamic response function, our hypothesis was well, that should actually depend on perfusion pressure and therefore autoregulation. And so the way we say this is, okay, if autoregulation is intact, we can use this general description that is heavily used in fMRI as well, assuming how the shape of of the hemodynamic response function, and outside that range, we don't really know what it is, and we want to know. Now, my lab is not only has quite a few different optical instruments available, but we're uniquely positioned to ask any of those questions because we have amazing clinical collaborators that helped us, or collaborators that helped us develop a preclinical model, in this case, non-human primates, where we can test a lot of those questions. And so the animal model we have available is such that we can change intracranial pressure by putting fluid in the ventricle by the means of a catheter. We have a pressure sensor to actually measure intracranial pressure, an A line for blood pressure, so we really have perfusion pressure, and we can place the optical sensors on the head, so we measure blood flow, oxy and deoxyhemoglobin, and then also with EEG, we measure the neuronal activation. And so the way we change intracranial pressure is, as I said, we put more of this fluid in the ventricles, and fluid is saline, and so by just lifting or lowering this bucket of saline, we can very controllably change intracranial pressure. And so we typically go from anywhere of 5 to about 30 millimeter mercury, and can step up. And so just to give you an example, so we measure ICP, blood pressure, and perfusion pressure then, so the difference between the two. And then optically, we measure oxy, deoxy, and blood flow, and we can also vary and induce oscillations in intracranial pressure or blood pressure. We're doing both, and we can look at transfer function analysis and the typical autoregulatory questions that that people are asking. So just to give you an idea of one thing that we have done is we, as a side goal, we had some animals that were anesthetized with isoflurane, so therefore impairing autoregulation versus fentanyl, which is known to maintain autoregulation. So we have actually shown that. So we measured cerebral blood flow in animals with isoflurane versus fentanyl, and you can see, so that's blood flow measured with diffuse correlation spectroscopy, and you can definitely see that perfusion pressure was very low in the isoflurane animals versus maintaining autoregulation in the fentanyl monkeys, except for when we change perfusion pressure very much outside that range. And I do need to hurry a little bit. And we also calculated pressure reactivity index, showing again that the pressure reactivity index is somewhat negative with the fentanyl monkeys, and never reached negative values in the iso monkeys, again showing autoregulation impairment. We do have a whole lot of data available where we could do more transfer function analysis and such. We just haven't done it yet. But let me just jump to the data that we have on non-invasive ICP estimation, which is based on the idea that the cardiac waveform in intracranial pressure has specific meaning, like the shape of the pressure waveform goes hand in hand with the baseline, with the amount of intracranial pressure. And we said, well, the same should be true for blood flow as well, which is actually an idea that we came up with more or less at the same time as Turgut's lab. So I don't know if he will mention some of that later as well. But it's, so we said, all right, we have low and high intracranial pressure. And then we said, well, what about the blood flow pulses that we measure in the microvasculature, that we optically measure non-invasively? How do they look like? And just to give you an example, here on the left, the orange curve is cerebral blood flow at the heart rate, at the cardiac pulsation, when intracranial pressure is elevated, versus the blue curve is at low intracranial pressure. And so what you see is there are morphological differences, especially the percussion wave first peak is reduced in magnitude whenever intracranial pressure goes up, which is, even though the magnitudes and shapes are completely different in intracranial pressure, but which is the same here in intracranial pressure as well. So the idea was, how do we link our blood flow measurements to intracranial pressure? And so the way we did this was, say we know there are certain features in those waveforms at low pressure and at high pressure. And those features we can describe. We can say, how wide is that cardiac waveform? What's the area under the curve? And how does the waveform look like? And so the way we then describe this is by saying, well, we can use regression forests, which is really a form of machine learning, but it's feature extraction. And so we can use a subset of the data for training and then a different subset of data for testing, and then say, well, what is the correlation between our invasively measured pressure and non-invasive pressure? And the correlation was pretty good. So what that allowed us to do was to then say, well, what's the difference? What's the Plante-Altman plot? And we have about a 4 millimeter mercury accuracy in the animals. So that's across 5 animals over many different hours. So we have thousands of individual pulses. But the accuracy was really stunning. And so we can do something like tracking of intracranial pressure over time as well. And so we're taking those systems now into the clinic, and we say, all right, just put an optic sensor on the forehead, measure every time the heart is beating, measure the blood flow, cardiac waveform change, and then we can assess intracranial pressure. And what intracranial pressure will allow us to do then is to ask about cerebral perfusion pressure, which gives us a more detailed understanding, hopefully, about cerebral autoregulation. So that's just one aspect of this whole idea. But then we also asked, so does autoregulation impact neurovascular coupling? So we measure neurons with EEG, the vasculature with near-infrared spectroscopy, autoregulation in terms of Larsen's curve with the optical sensor, diffuse correlation spectroscopy. And so the setup, I'm just going to rush through this. The setup is very much the same. In the animals, we change intracranial pressure based on fluid. We can vary, therefore, cerebral perfusion pressure. And we put EEG electrodes on the visual cortex as well as optical probes on the visual cortex. And then we give the animals a visual stimulus, which is consistent of rotating checkerboards. And so the stimulus design is that for about 5 seconds, we have this flickering of this rotating checkerboard, followed by about 10 seconds of rest. So it's a very typical block design used in fMRI as well. And then we said, all right, on the Larsen's curve, how does the neuronal activation as well as the vascular functional activation change with perfusion pressure? And so we step up here. Just again, this is our optically measured Larsen's curve with DCS. Again, we change perfusion pressure quite a bit based on intracranial pressure and blood pressure. And then we said, okay, can we, what you're going to see here is the data of the neuronal response and the vascular response as a function of perfusion pressure. So anything above about 100 is what we found is impairment of autoregulation. So we start out with what is known as a pretty normal looking visual evoked potential. And as soon as perfusion pressure goes higher, you start seeing changes, and the same is true for the hemodynamic response. Now, I was making a big deal of neurovascular coupling. What I'm showing you here is neuronal responses and vascular responses. Both of them change with perfusion pressure, but I really wanted to know what is the coupling between them, because that's much more important. And so again, a reminder, what we're calculating is the hemodynamic response function. And so the way we do this is, say, we take the EEG, we take the NIRS response, and we convolve the two, and we come up with this hemodynamic response function, and which we can evaluate back onto the signals. Now I'm really going to stop. So let me just jump to the conclusion, which is what we have found is that the hemodynamic response function, so therefore neurovascular coupling, is maintained, looks normal for about 60 to 100 millimeter mercury in our animals. And below that and above that, the response function itself changes. So that really means that the coupling between the two is either impaired or at least altered in a way. So if I overlay that onto Larsen's curve, whenever there's a plateau, the coupling stays constant and the coupling changes outside the autoregulatory range. In conclusion, what I've told you is we can calculate intracranial pressure quite accurately, which will allow us to say something about perfusion pressure, even in patients or healthy volunteers that do not have an ICP probe placed. And my lab is trying to figure out what exactly changes, where is the coupling change in neurovascular coupling coming from in terms of intracranial pressure changes. We're translating all of those optical methods to the clinic right now. So we're looking at traumatic brain injury, hydrocephalus patients, mainly because of ICP probes that are available. But we're moving towards stroke as well and other diseases. So with that, I would like to thank my lab, outstanding students and clinical collaborators, and my funding sources. And I gave myself about 30 minutes, so I'm a couple minutes over, but I'm more than happy to take any questions if there are some.
Great. Thank you very much, Jana. Fascinating story. Expect an email from me soon. And I see that Caroline has on top of that stolen my question. So I'll go with Caroline's question. So really interesting talk. Thank you, Jana. Can you comment on the effect of a skin contamination on NIRS measurements, and whether some NIRS devices approaches are more affected by this? And are there similar issues with the DCS?
Yeah. So let me take the second question first, which is, are there similar issues with DCS? Yes, absolutely. As soon as you shine light through the skin, you're influenced by the skin. Now there are ways around this, as in, for instance, like with the ICP extraction, the waveform itself is different in the skin versus the brain. Like we have arm measurements versus brain, so you can differentiate there. But the more classical way of dealing with this is a form of regression. So put short sensors, short distance sensors on the head and measure the skin itself to regress out the signal from the brain signals themselves. The more fancier instruments that I was talking about in terms of time domain, for instance, time domain instruments, and even frequency domain instruments, can remove superficial layers in another way by saying, okay, you immediately measure as part of it that comes from the skin. So all of those approaches, all of the optical devices are influenced by the skin, but there are many different ways of removing that. Yeah.
Great, thanks. Question from Dr. Schondorf. One group that may be of great interest may be those patients with headache owing to spontaneous intracranial hypotension. Also, those patients with idiopathic intracranial hypertension.
The intracranial, the idiopathic intracranial hypertension is something that I've talked about a lot with clinical collaborators and such. And we're starting now. So this year, the work was in animals, but we have now set up essentially a program for at least 100 patients in adults to really quantify the ICP extraction. But for that, we need the ICP measurement for now. But then once we have really validated the accuracy in more detail, then And again, also coming from Turgut's lab, similar efforts, then we can really ask the interesting questions like the idiopathic ICP, intracranial hypertension. And
a question from Stephen Payne. Many thanks for a very interesting talk. Could you comment on why you see an undershoot in the response when you move away from the plateau region of the Lassen curve?
Not yet. It's a brilliant question. So I don't know why I see the undershoot. So people have hypothesized, even from the fMRI community, and like using balloon models and then trying to come up with ideas of why the hemodynamic response function looks the way it does. And like even coming from my postdoc lab with Sergio Fantini, we've done a whole lot of modeling. The reality is I don't know exactly where it comes from. So it's very qualitative for now. I have a grant in right now that is under review this week that will hopefully help me to answer questions more quantitatively. So we're doing small animal measurements to really try to understand what the undershoot means, what the change in the coupling, where it comes from. Is it neuronally driven, or is it because we just put so much pressure on the vasculature that maybe oxygen extraction is changing? So I don't know, but it's a brilliant question that hopefully in a couple of years I'll have an answer to.
Yeah. I think the next one was similar to the latest about the initial undershoot. So if there is no other question, let's move forward with the other speaker, and you may still check in the chat feature. I'm sure people will continue asking questions. Well, thank you very much for a nice presentation.
Thank you. Yeah, sorry guys that I'm running late, like guys as in the following speakers. But let me introduce our next speaker, who is Thao Pham. She's a PhD student in Sergio Fantini's lab, focusing on near-infrared spectroscopy and autoregulation quantification. And so Fantini's lab has really been working on modeling the hemodynamic responses in the brain, especially in the context of autoregulation, and they came up with what is called coherent hemodynamic spectroscopy. So Thao, the floor is all yours.
Thank you, Jana. Thank you, the CARNET, for your invitation. Hello, everyone. My name is Thao Pham. I'm a PhD student with Sergio Fantini and the DOIT lab. And today my talk is about diffuse optical methods and coherent hemodynamic spectroscopy for the assessment of cerebral autoregulation. So this is the outline of my talk today. First of all, I'll talk about the cerebral hemodynamic oscillations induced by the oscillation in systemic blood pressure, and how to use this information to access cerebral autoregulation, what have been done with transcranial Doppler ultrasound, and our approach with that use NIRS. And then I'll talk about briefly about our model coherent hemodynamic spectroscopy, or CHS. And then one of the questions for Jana previously is about the issue of the contamination of extracerebral tissue into the optical measurement. So we also address this issue in this presentation. Then we also have some approach as a solution for this issue is to use the frequency domain NIRS phase measurement, and also our recently proposed method called the Diffuse method. And then I'll talk about some application to the measurement in human brain. For example, to measure the cerebral hemodynamic oscillations with different measurement techniques, and measure cerebral autoregulation during normocapnia and hypercapnia. So as everybody here is already familiar with, so people use transcranial Doppler ultrasound to measure dynamic cerebral autoregulation. And transcranial Doppler ultrasound usually give us the information of global measurement of cerebral blood flow velocity. And here I show here in a famous paper of Aslit that show the perturbation in arterial blood pressure that cause also the perturbation in cerebral blood flow. And people measure like how fast the blood flow measurement recover from the change in blood pressure to indicate cerebral autoregulation measurement. When checking in literature, we also found that so there's several studies that look at the phase relationship between blood flow and blood pressure as the indication for autoregulation. For example, in this study, they look at they measure blood pressure and blood flow velocity during normal capnia and hypercapnia, and look at the spontaneous oscillation of CBFE, blood flow velocity, and blood pressure in several frequency range. For example, here I show it in very low frequency range from 0.02 to 0.07 hertz, and low frequency range from 0.07 to 0.2 hertz. And this show that the phase relationship between CBFE and APP during hypercapnia is less positive compared to those measured during normal capnia, and is an indication of impaired autoregulation due to hypercapnia. So what I want to emphasize is that so this study, they look at the spontaneous oscillation of cerebral blood flow induced by blood pressure, and they look at the time frequency analysis of those oscillations, and they apply the coherence threshold to make sure that they just get those cluster of oscillation that pass to coherence. And our study also have some similar idea to look at the time frequency analysis. So another famous study is Reinhardt paper that look at the induced oscillation in cerebral blood flow by oscillation in blood pressure. So here in this study, they look at patients with unilateral obstructive carotid disease, and the patients perform paced breathing at 0.1 hertz. So that caused the oscillation in blood pressure, and thus it caused the oscillation in cerebral blood flow. Sorry. Something's wrong. Okay, so by looking at the phase difference between cerebral blood flow and blood pressure, they see that the phase difference for the obstructed size of the forehead has less positive phase for CBFE and blood pressure that indicate the impaired autoregulation. And this paper also look at the NIRS measurement of oxy and deoxyhemoglobin. And so this measurement, they look at the phase difference between deoxy versus oxy, and they show that for the obstructed side, the phase difference between deoxy versus oxy is more negative, and is an indication of impaired autoregulation. So I want to emphasize that these measurement or optical methods in general can provide a non-invasive and continuous measurement that is similar to transcranial Doppler ultrasound. But the advantage of optical measurement is that it can provide a local assessment of cerebral autoregulation in the microvasculature scale. So for our experimental protocol, we have several ways to induce oscillation in blood pressure. The first way, as you can see here, is to have the pneumatic tie cuff oscillation at a certain frequency, for example, at 0.1 hertz, that causes the oscillation in blood pressure. The second way is having the subject perform paced breathing at certain oscillation at certain frequency, and that's also caused the oscillation in blood pressure. And the third way to look at is the spontaneous blood pressure oscillation during baseline. And in the zoom in plot over here, you can see that the oscillation in blood pressure caused the oscillation in oxy and deoxyhemoglobin measured by NIRS. And the phase, relative phase between oxy and deoxy is the main focus of our study. So to quantify the coherence between NIRS measurement and blood pressure, we employ wavelet transform to look at the coherence. So here I show the coherence map of oxy versus blood pressure as a function of time and frequency. And you can see that those yellow cluster over here indicate that those region that has high coherence value. And then we perform the same analysis on random numbers to generate the coherence threshold and apply the coherence threshold on this analysis over here. So here you see that this is the significant coherence map of oxy and blood pressure. And those black cluster indicate that those that pass the coherent threshold. And here we show the temporal coherence as a function of time, a temporal coherence at 0.1 Hertz. And the pink line over here indicate the coherent threshold that generate from surrogate random numbers. And I just want to mention that the idea of generating the coherence threshold is also very popular in the field of transcranial Doppler ultrasound. After looking at the significant coherence between deoxy versus blood pressure and oxy versus blood pressure, we select those regions that like mutually coherence for both NIRS measurement versus blood pressure. And here I show the transfer function analysis that generate the amplitude ratio between deoxy versus oxy and the phase difference between deoxy versus oxy for those significant coherence region. And for the region of interest, if we take the average of these clusters over here, we can get the average phasor that is indicated by those vectors over here. Like here I show it, those average region for spontaneous oscillation, curve oscillation, and pace-breathing oscillation. And the amplitude of these vectors over here indicated the value of the amplitude ratio. And the direction of these vectors indicate the phase difference between the oxy versus oxy. So our group proposed the coherent hemodynamic spectroscopy model to relate the quantity that measured by NIRS, which is deoxy and oxy, to the underlying process that happen in the brain, such as blood volume, blood flow, and oxygen metabolic oxygen consumption. So this model has the input as the oscillation in blood volume, oscillation in blood flow, and metabolic rate of oxygen. And the output is the oscillation in oxy and deoxy. And the model is defined by these model parameters, such as the blood transit time in the capillaries and venous compartment, and the baseline blood volume fraction in the arteries, capillaries, and venous compartment. So the idea of the model is that we can see what happened during the change in autoregulation effect that affect the phase difference between CBF and blood pressure, and relate that to what happened in the phase difference between deoxy and oxy. So you see here that from poor autoregulation to effective autoregulation, the phase difference between CBF and APP is become more positive. And with that, when we look at the phase difference between deoxy versus oxy, we see that the phase difference is less negative, and it's also the phasor ratio also have the greater amplitude ratio. So before going into several applications on human brain, I just want to emphasize that optical measurements in general have a key issue that has the contribution from superficial extracerebral tissue. So you see here that the light that comes from the light source into the optical detector in the measured tissue, it has to pass the scalp and the skull tissue, and unfortunately, For this configuration, the high sensitivity region of the measurement is on the superficial tissue of scalp and skull. And there are several methods to minimize or correct for the extracerebral tissue contaminations, and I list a few of them here for reference. So our group will propose some methods to also to try to minimize or correct for the extracerebral contamination. But before that, let's look at like what will the hemodynamics look like for the scalp and the brain tissue. So we perform an experiment that have the subject have the pneumatic tie cuff oscillation at a certain frequency. And we look at NIRS measurement at different sort detector distance. And the short distance will be mostly sensitive to the scalp tissue that have that we expect to have poor autoregulation. And the long distance is sensitive to both scalp and brain tissue, and the brain tissue is expected to have more effective autoregulation. So this is the result of this study. So if we look at the amplitude ratio of the oxy versus oxy, we see that it has the initial decrease, and then later it will increase with short detector distance. And the phase difference between the oxy versus oxy, it become less negative over the short detector distance, and is an indication that so as we go to longer short detector distance, the phase become less negative, and is corresponding to the greater autoregulation effect, and so it's more sensitive to the brain. So with that, we also look at several other approaches to correct for the extracerebral contamination. So here I show the sensitivity map of several approaches. And the first one is the single distance intensity, which is what has been used in our study before. So you see this is the banana shape that have high sensitivity close to the superficial tissue. One approach is to look at the phase frequency domain phase signal instead of intensity signal. So I just want to describe quickly that the frequency domain NIRS has the intensity that of the light that we use is oscillate at certain frequency, and then the detected light also oscillate, and we look at the phase difference between the detected light and the input light, which give us the phase information. So by using the frequency domain phase information, we also have a banana shape of sensitivity region, but it's go deeper into the measurement, the measure of tissue as compared to the single distance intensity. Another approach is to use something called a dual slope configuration that require two source and two detectors. And so you can see the dual slope intensity over here. It has the sensitivity region that is localized between the two detectors. And the sensitivity region of dual slope intensity is relatively deeper as compared to single distance intensity. And finally, if we look at the dual slope phase measurement, it has the sensitivity region deep into the tissue and also localized, more localized as compared to other methods. So in one study, we look at the oscillation induced by tie cuff oscillation and pace breathing at a certain frequency. And then we look at the measurement at different sort detector distance. And this is the result of the study. So here we report the measurement of the oxy versus oxy at single distance intensity measurement, which is indicated by the purple lines over here, and single distance phase measurement is indicated by the green lines over here. And we see that the single distance phase measurement gives greater amplitude ratio for the oxy versus oxy as compared to single distance intensity. And it also gives us less negative phase value as compared to single distance intensity. And in general, you can see that the single distance phase measurement show weaker dependence on the source detector distance as compared to single distance intensity. And the takeaway message from this study is that single distance phase is more sensitive to the brain with greater autoregulation efficiency than single distance intensity. And you can see that the sensitivity to the brain even start at very short detected distance. And in another study, we look at the pneumatic type of oscillation in healthy subject, and we use the dual slope probe. So I showed this configuration before with two source 1 and source 2, and two detectors, detector A and detector B, and it can give us two measurement of single distance with at 35 millimeters or detector distance, and one dual slope measurement. And this is an example of this the result of this study. So this the plot on the left, so the frequency domain is intensity measurement, and the one at the right show the phase measurement. So the blue arrow indicates single distance intensity, single distance measurement. The yellow arrow indicates dual slope measurement, and the single slope measurement is somehow similar to dual slope measurement. I didn't mention in this presentation, but the average of the two single slope will give us the dual slope. So for intensity measurement, you can see that the amplitude of the single distance intensity is very small as compared to the amplitude ratio of the oxy versus oxy measured by the dual slope measurement. But if we look at the phase measurement, both single distance phase and dual slope phase has higher amplitude as compared to the single distance intensity. And for the dual slope intensity and the phase measurement in general, they have less negative phase as compared to single distance intensity. And on phase measurement is around negative 200 degree, which is an indication that it is more sensitive to the brain autoregulation. So yeah, so the conclusion of this study is that dual slope is more sensitive to the brain with greater autoregulation as compared to single distance intensity measurement. And finally, so we just recently have a study that had the subject with oscillation induced by pneumatic tie cuff at 0.07 Hertz with the dual slope measurement for the optical nearest measurement. And for this subject, we have the subject breathe medical air to induce normal capnia, and air mixed with 5% CO2 to induce hypercapnia. So we have one pneumatic tie cuff oscillation during normal capnia, and one is during hypercapnia. So in this study, instead of looking the phasor ratio between deoxy versus oxy, we use the information measured by NIRS to translate into the phasor of a CBF. So by using the CHS model, so with the CHS model, we can correct for the oscillation in CBV and also the contribution of the blood transit time in the capillaries and venous compartment to the measurement of CBF. So here is the result of this study. So you can see here that I show, instead of deoxy versus oxy, I show the ratio of CBF versus normalized blood pressure. It's lowercase APP over here. And I show the arrows indicate the measurements for dual slope intensity, which is the purple arrows, and dual slope phase, which is the green arrow. And you can see that the dual slope phase measurement show a smaller amplitude ratio and more positive phase as compared to the dual slope intensity at normal capnia. And then if we look at the hypercapnia measurement, which indicated by those dashed lines over here, the hypercapnia measurement show less positive phase as compared to all of the normal capnia measurement. So the conclusion is that the dual slope phase measurement seems to give more sensitivity to the brain with greater autoregulation as compared to dual slope intensity. And also the CBF we got from frequency domain near CHS is sensitive to the change in autoregulation during hypercapnia. So this is the end of the presentation, and as a summary, we show that this measurement of blood pressure induced several hemodynamic oscillations can show some sensitivity to several autoregulation efficiency. We can either use the indirect measurement of deoxy versus oxy, and we have demonstrated that these measurements show some sensitivity to scar versus sprain autoregulation efficiency. And also initial results show that the CPF measured by frequency domain near CHS is sensitive to autoregulation efficiency during hypercapnia. I also summarized some efforts of our group to try to improve the sensitivity of frequency domain near measurement to deeper tissue. For example, we can use the phase single distance measurement that show more sensitivity to the deep tissue as compared to single distance intensity. Or we use the phase dual slope measurement. And I just want to mention that the dual slope approach can also show weak sensitivity to motion artifacts, instrumental drift, and optical couplings. With that, I would like to acknowledge the DoIT Lab and the support. Our studies are supported by NIH and BSF. Thank you for your attention.
Thanks a lot, Thao. I must start my video. Are there questions from the audience? There's at least one from Pat that said, so very interesting, thank you. Have you thought about doing similar analysis using repeated squat stance and oscillatory lower body negative pressure to force blood pressure oscillations? So that's part of the first part of your talk.
Yeah, so our group thought of a lot of several approach for inducing the oscillation in the systemic blood pressure. And so the reason we try to use the oscillation is that it causes less motion artifact to the optical measurement.
Which makes sense, yeah.
Yeah.
Are there other questions from the audience? Feel free to just unmute yourself and ask them, or I'm scrolling through the chat.
I think there's a question about what is the level to hypercapnia.
Oh, I missed that. Yeah.
Okay. Yeah. So I understand that. So this question is about how to induce hypercapnia. So we have the subject breathed medical air mixed with 5% CO2 to induce hypercapnia.
Okay, sorry, this is Dawn Wolfgram. I had asked that question. So what level of CO2 did they get up to with the 5%? Like PCO2, sorry, that was measured, or was that just not measured?
Yeah, so that's a good question. I don't have the blood of entitled CO2 over here, but I think it's like around 50 something.
Okay, thank you.
Yeah, thank you.
Great. If anyone has other questions for Thao, please just write it in the chat, and she will, I'm sure, be glad to answer them over the chat. So maybe with that, let's move on to Wes Baker. Wes, you wanna?
Yeah, sure.
So Wes is an assistant professor in neurology at the Children's Hospital in Philadelphia, and got his PhD from Penn in physics, I believe, and is going to tell us about autoregulation measurements in ECMO and cardiopulmonary bypass. Go ahead, Wes, it's all yours.
Yeah, thank you, Jana. And I'm very grateful for the invitation to speak today. As Jana said, I will be giving a discussion of some of our preliminary measurements of optical assessment of cerebral autoregulation during ECMO, cardiopulmonary bypass. I'll mostly focus on our measurements of autoregulation in infants on ECMO. Time permitting, at the end, I have a few slides about cardiopulmonary bypass in the piglet model. So here's a picture of a baby on ECMO at the Children's Hospital of Philadelphia. ECMO essentially uses a mechanical pump and oxygenator to maintain perfusion and oxygenation while resting the injured lungs and or heart in very sick infants. In particular, usually like a large artery and a large vein are cannulated. So typically, outflow from the jugular vein is directed to the pump, which pushes flow through an oxygenator and into the carotid artery. So in this picture on the infant, this is the flow from the jugular vein going to the pump, and this is the incoming flow going into the carotid artery to perfuse the body. So one important question in ECMO is what should you set the pump flow rate to? Presently,
hey Wes, I'm sorry to interrupt you, but your audio keeps breaking up. I don't know if you can change anything.
Yeah, sorry about that. Hopefully it will get better. I'll continue now. If it still gets really bad, let me know and I'll try to switch. But yeah, so the pump flow rate presently is set by patient weight, and then it's adjusted based on systemic vital signs. The problem is that systemic vital signs are not always a direct assessment of what brain health is. And notice that in this case, there is no brain monitoring on the infant to directly see what is cerebral perfusion. So there's a substantial portion of patients that perhaps are likely not receiving adequate oxygen delivery due to the pump flow rate not being set correctly. And that could be a contributing factor to the neurological morbidity of approximately 50% observed in ECMO survivors. So one of our goals is that clinicians are presently blinded to how the systemic vital signs are influencing the brain. And we hope that optical monitoring can help fulfill that clinical need. And in particular, especially for this talk, we're going to look at impaired cerebral autoregulation as a risk factor. So as you know, autoregulation mechanisms work to maintain adequate cerebral blood flow to the brain. But during ECMO, it's definitely possible for cerebral autoregulation to be impaired. And then when that's the case, there's a risk of hyperemia or ischemia, both of which can cause strokes. So for the clinician to better manage this risk factor, like a key first step is they just have to have the ability to detect it in the first place. In order to better manage a risk factor, you have to detect it. So we're focused on basically techniques to non-invasively detect impaired cerebral autoregulation during ECMO. And we tried doing this using the optical technique of diffuse correlation spectroscopy that Jana talked about. Well, non-invasively monitor cerebral blood flow during ECMO. Then we used cerebral blood flow and mean arterial blood pressure correlations to characterize regulation, as I'll talk about. Yeah, so Jana already introduced this, but very briefly, diffuse correlation spectroscopy uses intensity fluctuations of tissue scattered light from source to detector to probe blood flow. The higher the blood flow, the more rapid the intensity fluctuations will be at the detector. And you can quantitatively characterize these with an intensity correlation function.
Last interruption, I promise.
Oh, no, sure.
Your audio is better when you don't lean forward. If you just stay back and you don't move, then it'll be great.
Okay, thanks. Yeah, I'm not sure what's up with this today, but yeah, I'll try that. Yeah, so for DCS, you just fit the decay of this intensity autocorrelation function to a correlation diffusion model, and you can extract a blood flow index, which has been shown to be proportional to blood flow. So in this study, we monitored 9 neonates on ECMO at the Children's Hospital of Philadelphia. In particular, 40-minute cerebral blood flow monitoring sessions were performed daily during these induced ECMO pump flow changes, plus or minus 10%. So the idea of these pump flow changes is actually to induce mean arterial pressure changes to increase the signal to noise of the autoregulation assessment. And this is just kind of a reminder of the ECMO schematic here, where during ECMO you have the ECMO pump flow in parallel with cardiac flow. And then both work together to influence the mean arterial blood pressure as well as the cerebral blood flow. And as you know, the relation between these is how we're looking at that to assess autoregulation. And in particular, during each 40-minute session, we used a moving 2-minute Pearson correlation between cerebral blood flow and MAP. So to assess autoregulation this way, it does require physiologic variation in mean arterial pressure. And the way to think about this is that across a given temporal period, changes in the MAP, they can consist of physiologic variation, but then also measurement noise. So what happens is if you have minimal physiologic variance in MAP, then the change is just going to be dominated by measurement noise, and that's going to be random and intrinsically uncorrelated from the cerebral blood flow. So the Pearson correlation will basically be reflective of this, which averages to 0. So if there's no physiologic variance in MAP, then the Pearson correlation will be 0, regardless of whether autoregulation is intact or impaired. So you really need this to be present for it to be meaningful. So to address that, we used two strategies. And the first, as I already mentioned, was to actually change the pump flow by plus or minus 10%, with our hypothesis that that would induce corresponding changes in pressure. But then what we also did is that when we were using our moving 2-minute window to compute the correlation between CBF and MAP, we specified a requirement that the MAP within the window had to actually significantly change for it to be used in our assessment. So what that means is that for each 2-minute window, we computed the maximum MAP minus the minimum MAP in that window. And we said that that had to be greater than 3 millimeters of mercury. If it was, then we'd use the Pearson correlation in our autoregulation assessment. If it was not, we'd ignore it. And this is just an example of an autoregulation assessment we did during a single measurement session in an ECMO patient. So this is the raw MAP data versus time across the 40 minutes. This is the raw cerebral blood flow normalized to baseline versus time. And then these bottom two plots are where we did the 2-minute moving window with maximum overlap. So this third plot is the delta MAP I just talked about, meaning the maximum minus the minimum MAP for each window. And we required that it had to exceed this threshold of 3 millimeters of mercury to be used. So this is the delta MAP. And then this bottom plot here is the Pearson correlation between cerebral blood flow and mean arterial pressure. So what you can see is that when the delta MAP exceeds the threshold, you get a very strong correlation, close to 1. However, when MAP's not changing as much, the correlation is much closer to 0. So in our method, we averaged the Pearson correlations only for those windows that exceeded that threshold. And we got 0.9 for the autoregulation assessment during this session. If you were to average all the windows, you get a correlation coefficient substantially less at 0.5. So this patient did have impaired cerebral autoregulation. And if you use this delta MAP threshold, we hypothesize that you'll be more sensitive to detecting impaired autoregulation because it will remove kind of these zeros when the MAP's not changing. So the other question I had before I get to our main results is just, do the pump flow changes actually alter MAP? And yes, they do alter MAP, but the alterations are small. I'm just showing one example here where this is the pump flow versus time during our monitoring session. It's changing by minus 10% and then plus 10%. And then this is the corresponding mean arterial pressure data. These plots on the right are just the averaged data at each pump flow level. So for instance, this map point here corresponds to the average during that level. This point here corresponds to that level, and so on. And then finally, this bottom plot is just the difference between adjacent pump flow levels in the map. So in this subject, plus or minus 10% change in pump flow changed MAP by 1.6 millimeters of mercury on average. So not very much. And similarly, if you do a box plot of the magnitude of these changes across every single measurement, we found a similar average. Plus or minus 10% change in pump flow will change MAP, but only by about 2 millimeters of mercury. So a pretty small change, but it still could be helpful if you have the ability to do this. By the way, this corresponds basically to about a 4% of the mean, the magnitude of that change. Okay, so now that we've talked about how we characterize autoregulation with the delta MAP method and the pump flow changes, I was just interested in comparing to the systemic parameters that clinicians typically rely on for setting the ECMO pump flow. So the first thing we looked at was just the average MAP during the measurement session. So there's actually 21 measurement sessions, and I grouped it by MAP terciles here. So the main thing is there's actually no difference in our autoregulation assessment between the MAP tercile groups. So at least in terms of the absolute MAP, it's not so reflective of, it's not predictive of autoregulation. And there's a wide variability, I guess, within each group. The same result was observed for the pump flow. So now this is just looking at the average pump flow during the measurement sessions and grouping it into terciles. And there's, again, no significant difference in autoregulation and pump flow. However, interestingly, now if we look more at pulse pressure, the autoregulation now does differ across the pulse pressure terciles. So pulse pressure is the difference between systolic blood pressure and diastolic blood pressure. So we took the average of this during our monitoring session. And this can be used as kind of a metric of cardiac function, because remember, during ECMO, the pump flow, which is constant, this is steady state, is in parallel with the cardiac flow, which is pulsatile with the heart rate. So the larger this pulse pressure is, the more the cardiac flow is that's pulsatile is contributing. And so basically, if the heart's not working at all, the pulse pressure will be close to zero. If the heart's functioning well, the pulse pressure will get much higher. And here, specifically between these two groups, autoregulation was impaired for the low pulse pressure group, and it was better in this pulse pressure group. And note that the MAP levels between these groups are similar. There's no difference in mean arterial pressure between these groups. But then from pulse pressure, you can see the ones with low had impaired autoregulation, and the higher pulse pressures had intact autoregulation. So this is just an interesting result that suggests that the autoregulation during ECMO could be sensitive to blood pressure pulsatility. And important future work will be to show if you induce a pulsatile component in the pump flow, can that help to preserve autoregulation in what would otherwise have been impaired? So this is pilot data that motivates that future study. And then I just want to mention also the importance of our delta MAP threshold. So this association we saw between autoregulation and pulse pressure when using the delta MAP greater than 3-minute threshold is lost if you use all 2-minute moving windows. So you lose the effect here, even though it's trending the same. So this is just evidence that if you do use a delta MAP threshold in your windows to compute R, you're more sensitive to impaired autoregulation. So this is pilot data supporting that hypothesis. And then I'll also just give some cautionary data in terms of, relating autoregulation to outcome. So I'm just showing now longitudinal data for 2 subjects in the study. The subject on the left was weaned off ECMO on day 6. So the day after this, the subject survived and was successfully weaned off. And these are showing our daily measurements across the 5 days of ECMO. So this is pump flow, mean arterial pressure, pulse pressure, and then the autoregulation index. And what you can see here is that the autoregulation is mostly impaired across our measurements in this subject, even though it survived and was successfully weaned off a day later. Then this is just another example. This subject ultimately died after 26 days of ECMO. We were able to monitor the first 4. And in this subject, our autoregulation index shows intact autoregulation. The correlations are close to 0 between MAP and cerebral blood flow, though the subject still died and still on ECMO for 26 days. So this just kind of shows that there's certainly other risk factors besides just autoregulation in terms of predicting outcome, which is why you'll do like large studies in the long term. So to just conclude about this, diffuse correlation spectroscopy is a promising technique to monitor cerebral autoregulation during ECMO. It's similar to transcranial Doppler ultrasound, though it's not as sensitive to probe orientation. So it's actually, I think, better suited for prolonged monitoring if you want to monitor for a long period of time. Just that you just focus on the windows where there's significant MAP variation when assessing the autoregulation, as I talked about. We did show that autoregulation integrity did vary between individuals with time. So we did not see an association with systemic average MAP levels during ECMO, though we did see that a pilot association between low pulse pressure and impaired cerebral autoregulation. So future work would just, I think, be to directly compare the effects of pulsatile versus non-pulsatile flow in an animal model of ECMO to see, is it actually the pulsatility that's related to the improvement in autoregulation or not? And then also longer time period studies are needed with more patients too to better understand, longitudinal variations in autoregulation with medication and systemic change. So I'm close to out of time, so I'll just only spend a minute here, but we also looked at autoregulation the same way in an animal model of cardiopulmonary bypass, and that's used for heart surgery. So major heart surgery in infants with severe congenital heart defects does require cardiopulmonary bypass, and there's different perfusion management strategies during the surgery. And I'm just showing the 2 major ones here. They both involve deep hypothermia where the baby is cooled. But then in one strategy is circulatory arrest where then flow is completely turned off while they do the heart surgery to fix the heart. While the other is then selective cerebral perfusion, where the brain is partially perfused, but then flow to the rest of the body is turned off. And in reality, in surgery, like if they do selective cerebral perfusion, the surgery will take longer. So that's the drawback with the SEP case. So we were just interested in, do those strategies affect autoregulation differently? So we did this in a neonatal swine model of bypass, and we actually had four groups. So the control group did not receive hypothermia at all. It was just on bypass the whole time. Then we also had a deep hypothermic cardiopulmonary bypass group. This group did receive hypothermia, but then it remained on fairly high bypass flows. I mean, I'm showing a lower bypass flow here than controlled because it was necessary. Otherwise, the map got too big during the cooling. And then the other groups are the selective cerebral perfusion and deep hypothermic circulatory arrest that I talked about. So we did pump flow modulations before and after this intervention period to look at autoregulation exactly the same way as with ECMO. And here's what we found. And probably the biggest result is that there was a large variance in the pre-intervention autoregulation across animals. I'm separating them out by perfusion strategy here. So it's really not too much difference in like basically the absolute autoregulation assessments across the perfusion strategy. One, because there's a large variance even pre-intervention. But then we also just looked at the pre to post changes. So even though due to other factors, autoregulation differs across animals, like the intervention itself, how does that alter autoregulation? So we looked at the changes. And what we found is that for all the strategies except for the deep hypothermic cardiopulmonary bypass, there was no change in the autoregulation index between post-intervention and pre-intervention. However, for the deep hypothermic cardiopulmonary bypass, the autoregulation significantly increased, which suggests that maybe autoregulation became more impaired during this intervention. So this result may possibly be explained by like hyperperfusion in the deep hypothermic cardiopulmonary bypass group, where you induce deep hypothermia, but then the flow is still high. So you're probably hyperperfusing the brain. And that may be leading to like a worsening in the autoregulation mechanism is one possibility. So a future work for this is also needed a larger sample size to better understand this variance, especially, and as well as just a longer term study to track autoregulation changes during the post-intervention period. So I'd certainly like to acknowledge my team and collaborators, and please feel free to email me your questions, or I can take a couple of questions now. So thank you.
Thanks a lot, Wes. There are quite a few questions from the audience. I don't think we can get to all of them, but let me just start from the top. So Ronnie Panarai was asking, is the correlation the same as the MXA index used with TCD? Yeah, and maybe talk about differences there.
Yes, that's correct. So the correlation is the same as the MX index. It's just we're using an optical technique to measure cerebral blood flow instead. And the main advantage of the optical technique is I think it's a little easier to use that for prolonged monitoring, like if you want to monitor over many hours. And then the other thing that was different was the use of the delta map threshold to select windows, which we found actually helped improve our sensitivity to detect impaired autoregulation.
Right. So let me combine a couple of things here, because there's another question that says, what was the threshold of 3 millimeter mercury based on, and how many people, what was the percentage of ABB data that did not meet that threshold? But then also going hand in hand with another comment by Ronnie Panarai was about that R=1 suggests impaired autoregulation, but 0 really cannot be interpreted as efficient autoregulation because it could simply be based on noise. So I feel like the two are going hand in hand. So can you talk about the 3 millimeter mercury?
Yeah, I agree that the 3 millimeter threshold is arbitrary. We did try looking at some different thresholds to see if that influenced the result. So like, for example, if we used 2 or 4 millimeters mercury, that did not change the result in terms of the pulse pressure finding. You know, but if we use 5, then we lost too many subjects. So unfortunately, the pump flow changes did not alter MAP as much as we were hoping that it would. So we couldn't use really higher thresholds. So we kind of chose 3 as a compromise to try to ensure that MAP actually changed, but then also allow us to keep most of the patients. At the threshold of 3, we still lost 1 monitoring session. So we had 22 and we lost 1, so we had 21. But with that threshold, we at least were able to keep most of the data, but then it helps to kind of clean up, you know, contamination when you had just zero correlations for windows momentum change.
Maybe let's take one more, and then if you could just answer in the chat, maybe to everyone, so that other people know what the answers are. Up to you. One more question was, did you clamp other variables such as PCO2, which could have influenced cerebral blood flow during pump flow manipulation?
Yeah, that's a great question. So, like the longitudinal case examples I showed, PCO2 was fairly consistent across those days, so there wasn't too much difference there. But a lot of other things were done in the meantime in terms of medications, and that's why, like, we only did 40 minutes of monitoring, even though in principle you could monitor for much longer. Right. I think that will be important future work, is to really monitor for longer time periods, so you can kind of keep better track when, like, medications are given or ventilation settings are changed. You can see how that influences autoregulation. During the assessment itself, that was 40 minutes, that the PCO2 level remained constant.
Thank you, Wes. All right, we do need to move on to the last speaker. Thanks again, Wes, for a really great talk. So our last speaker for the session is Turgut Durduran from ICFO in Spain. So he's leading the medical imaging group there, one of the pioneers in diffuse correlation spectroscopy. Got his PhD from UPenn in physics, and the floor is all yours, Turgut.
Hello. First of all, thank you for the invitation. It's really a pleasure to be here. I suppose my slides are working fine. You can see them.
Yes.
Good. Well, also for Jana and Wes, you have really introduced all the methods very well, so I can just move on to results. Pretty much everything I'll show you today is with DCS, and some of them contain a little bit of time-resolved NIRS, basically. So I'm just gonna skip the description. So since the topic is autoregulation, and I normally don't give talks solely dedicated to autoregulation, I decided to be a little bit sort of adventurous here, and I put together very fresh out of the oven type of analysis that we are still hashing out from a couple of studies, and I will go through them to give you different ways we have started to look at autoregulation. So one study is this study on traumatic brain injury, which we are conducting with colleagues from Hospital Valdebron in Barcelona area, with Dr. Sauki and Dr. Poca. These are severe TBI patients, mostly sedated and basically artificially ventilated. And what they were interested in was to see if hyperventilation therapy, which is sometimes used in TBI patients to reduce intracranial pressure, leads to conditions such as misery perfusion or worsened autoregulation. So here I'll focus on the autoregulation part. So basically every subject was measured during baseline, and they were induced a hyperventilation, which was based on a certain drop of millimeter mercury in the arterial blood gas that was confirmed by gasometry. And this was kept in place for about 30 minutes. And then we looked at them at the recovery period. All patients were measured bilaterally on the frontal lobes, as you see in the photo, using TRS and DCS. And in order to do this, we took fast DCS data. So these were DCS data taken at about 40 Hertz. We also had time-resolved spectroscopy, so we have access to oxydeoxyhemoglobin and oxygen saturation. And by going through either a lab chart or MOBET, depending on which year we did this and what they had in the neurocritical care unit, we also synchronized everything fairly carefully with invasive ICP monitoring, the ventilator data, physiological monitors, and PTIO2 brain oximetry. So in particular, we'll be focusing on the ICP and MABP. And I should also say that almost all the data I show here come from two PhD students in my lab, Susanna Stagliabu and Jonas Fischer. And it's built up on about a decade of work by others, which I cannot acknowledge one by one here. So this is a typical data that we get from these patients. The first part, you see on different rows, you see intracranial pressure, partial pressure of oxygen in the brain, relative cerebral blood flow, relative oxygen extraction fraction, and relative cerebral metabolic rate of oxygen extraction, CMRO2. This last parameter is derived from NIRS and DCS together. What you see is that the hyperventilation in this particular patient within the two dashed lines caused a significant reduction of ICP. Also, PTIO2 was reduced in this patient, and CBF was also reduced with possibly somewhat visible reductions in the oxygen extraction fraction and CMRO2. So probably this patient didn't have any mesory perfusion. That's a separate study, separate analysis than what I will show you. But if you look at this data, and if you do the typical plot of plotting relative cerebral blood flow on the y-axis and cerebral perfusion pressure on the x-axis, and if we label different periods by different colors, we see that, for example, during the baseline, the sort of pinkish colored period, the two are practically not correlated to each other. The results are fairly flat. Whereas during the hyperventilation period, we see a significant correlation between the blood flow and the CPP, which disappears during the recovery period. This is a fairly typical data we get from these patients, and we don't yet know what it really means. We are trying to understand whether it's related to outcome, etc. Here's another case study. In this particular subject, you also got similar results. ICP went down, albeit much smaller amount. So the axes here are changing. So when data looks noisier, doesn't mean that it's necessarily noisier. You see the blood flow didn't really change much, or EF didn't change much, and CMRO2 didn't change much in this particular subject. You see that there are also abrupt changes that occur sometimes, which reflects itself in the whole patient physiology, such as those between about 4,000 to 5,000 seconds. And if you plot this person in the same way, okay, data is a bigger spread, perhaps a better denoising would be also useful here. But we also see a similar behavior. This is very typical. So we see baseline periods were flat, hyperventilation periods showed some type of a correlation. Again, as I said, the ranges were much smaller in this case, and the recovery periods were flat. So we are looking into this in details, but overall it sort of makes sense. If I look at this complicated looking graph from a recent paper, it's well understood that hypocapnia, especially as the severity increases, reduces the maximal vasoconstriction that's available to deal with elevated CPP, and reduces the range of the part in the typical Lassen curve where the autoregulation seems to be working and it's intact. So perhaps we are observing this. But since we have fast DCS data here, we can also look at other signatures of autoregulation. So Jana has already shown that fast DCS is able to see the pulsatile component of blood flow. Many fast DCS systems, such as those from UPenn and those from MGH, and I believe also from Jana's group, use softwares. In our case, we have developed hardware correlators shown in the inner picture in a large European project that allows us to stack many channels up to 32 hertz and obtain data at about 40, even at 100 hertz without compromising any part of the autocorrelation curve. And my PhD student Jonas looked at it in a couple of different ways on the same subjects. One of them was to do sort of a qualitative analysis. So he wanted to look at the to see if we can see spontaneous CBF response to natural fluctuations of CPPO arterial blood pressure. It's often stated that slow waves, sort of 0.05 hertz to 0.05 hertz, or 5 to 10 millimeter mercury in magnitude, are dominant in sedated and paralyzed patients. Do we see them in the CBF data? Maillard waves tend to be underrepresented in ventilated patients. Do we see their disappearance? And respiratory waves have very low signal-to-noise ratio in such things. So we'll look at that to see what we see. Also, he calculated the so-called CBFX index. In this case, he took 3-minute windows, binned in 5-second chunks, and shifted them by every 3 seconds, and calculated the Pearson correlation coefficient between BFI and either CPP or MAP. In the transcranial literature, this is called the MX. It's derived from the microvasculature, from the arterial major cerebral arteries. It has values -1 to 1. I think this audience knows it. Don't have to say much. Low values and negative tends to imply good cerebral autoregulation. There's literature saying that MX values greater than 0.3 indicate poor outcome. But for example, Bradley et al.'s work with laser Doppler, looking at the microvasculature, showed that higher thresholds would be needed for microvasculature, and CBFX from DCS, we don't yet know what to expect. We don't have thresholds, but we're just plotting them, and we are sort of showing that we see these things in our patients. So let's look at these so-called slow waves. These are 2 cases shown on the left and the right. It's the same severe TBI patients. What you see is that in the Fourier transform, that in the patient shown on the left, you see very significant and very distinct components in both ICP, ABP, and blood flow. And these things do not exist in healthy subjects. If you look at another patient, for example, you see much less of this. You see maybe 1 peak, and you see the same peak in the blood flow, and you see sort of dampened in the arterial blood pressure and the blood flow measurements from the brain. So we have to understand, quantify these, and relate them to the patient's well-being and other clinical status still. But the signatures are there. Also, if we take one of these patients, and if we quantify color code time, and if you plot blood flow index from the brain versus the cerebral perfusion pressure, we tend to see sort of different time periods bunched up together. We see, for example, the flat region in this patient after around 60 millimeter mercury CPP. We see that the 2 flat regions are parallel, the ones from the beginning, dark blue, and the red, light blue. And I just realized that my arrow points should have been pointing upwards in the color map, but on the bottom you see the time color coded. And if you turn this into a CBFX index, we see CBFXs which are very high at certain points. We see those that are low. We see fluctuations in this value. And we see that some of these are correlated with vertical bars that I have here, which correspond to the changing the noradrenaline given plasma lights, isoprotons playing with ventilator settings. So as I said, this is fresh out of the oven type of analysis, and we are trying to look into all these type of data to understand if these mean anything. These correlate with different drugs that are given to these patients and different things that were done to them. It's the same patient few days later in the second day of measurement. We see the same type of color coding, but we see this time that there's a significant period where the 2 parameters are just correlated with each other, resulting in very high CBFX indices, and early on periods where the things were flatter with a lower CBFX indices. So things appear to be evolving with time, evolve within one single session of measurement, and they are changing between day to day, where in some days we see flat regions, and some days in the similar CPP pressures, we don't see the flat region. So this was sort of individual data from severe TBI patients. So let me also show you something that we have been working on for many years on looking at group level observation of cerebral autoregulation impairment. And this we are looking at it in this sort of ischemic stroke many patients. So this is data that we have been working with colleagues from UPenn and the hospital in Barcelona area. And basically, we are trying to identify different parts where diffuse optics can be relevant in ischemic stroke management. And we're trying to look at biomarkers that would help us in the long-term prognosis and also identification of things that may be emerging at short time. This data is, as I said, covers many years and has many aspects. But what's common is this idea that John Detra from Hospital of University of Pennsylvania had. He said, look, people are trying to use orthostatic challenges as a way to change blood pressure as a possible biomarker of cerebral autoregulation. It's very complex. It's not just autoregulation that you'll be looking at it, but let's give it a try because it's non-invasive. You don't need the patient to collaborate with you, and patients already lie on movable beds. So we've been putting a lot of bilateral probes on these patients, and we do things like we keep them flat for 5 minutes. Then we tilt the head of bed position up to about 30 degrees for 5 minutes and bring them back to 0. And from this, we have been obtaining data like this. First 2 papers that with the data that we took at UPenn, what we found is that about 25% of severe ischemic stroke patients had what we call paradoxical responses. I should first clarify something. When we started the study, we expected the data to look like what you see on the red line, that autoregulation should take care of everything, and this small, soft orthostatic challenge shouldn't cause changes. But the deeper we dug into our data, even some MRI data with crazy rotating gantries, and also looking at some TCD data, it became very clear that you actually get a significant, but yet small change in the healthy subjects that looks more like the blue line when you elevate the head of bed position, even by this little amount, like 30 degrees. But what we were seeing is that in about 25% of these patients, especially on the ipsilesional hemisphere, the hemisphere where the stroke has occurred, we didn't see this decrease in blood flow, or we were seeing increase in blood flow, which we call the unexpected response. We dug really deep into this, but we were not really able to identify any significant patterns, partly as it will become more evident as we look into the data, because we were looking at the patients a bit late, after like 3 days after the stroke. It was, let's say, an opportunistic time window to look at them when their condition was more stable. As you may all know, I will just briefly go. Stroke is characterized by the so-called National Institutes of Health stroke scale. It's a scale between 0 to 43, and the higher the number, worse the condition is. So we dug very deep into this. I can give whole talks about this data. We didn't see very clear relationships. Another interesting parameter, however, is to look at what happened to these subjects 3 months after the stroke onset. This is sort of an outcome variable called modified Rankin scale. 6 implies death. 0 means the patient doesn't show any symptoms, and higher numbers imply different levels of disabilities. So when we look at the data sets that we were able to acquire in the early hours from stroke onset, less than 12 hours, ideally we should do it with less than 6 hours, but we had too few points. We ended up with 17 measurements. And what you see in this graph is we have plotted the same type of cerebral blood flow changes with respect to the modified Rankin scale 3 months after our measurement. What you see is that the so-called good outcome, modified Rankin scale of less than 2, show the typical response on the ipsilesional hemisphere. But once you get into subjects who are showing no changes or paradoxical changes, you see that they have tended to have higher modified Rankin scales. So this orthostatic variable, which may or may not be related to autoregulation, appears to show interesting, be a biomarker of the outcome in the long term, which we have to study better with better devices and have higher data sets. My former PhD student, Dr. Clara Gregory, was quite ambitious, and she contacted UPenn. Well, part of the data I already had in my hand, part of the data she didn't. So she put together a lot of data. She went through all the inclusion, exclusion criteria, differences in measurements, difference in devices, and ended up with 72 subject measurements from these 3 different studies performed in 2 countries, showed that the clinical and optical results were repeatable. There was a difference between ages, but as you look at newer and newer times, you end up recruiting older and older patients under these conditions, but everything else was similar. And she started sort of doing data mining in this data set. She wanted to look at, can she look at the earlier and earlier periods with larger numbers, and can she get new findings? And one of the interesting findings she found, and this is what we call the group level damaged autoregulation, was that if she looks at the ipsilesional hemisphere, not the contralesional hemisphere, it looks at the earliest data set that is viable in terms of size of subjects. This is less than 48 hours. What she saw is that the subjects who didn't return well to the supine position, to the original blood flow values upon going back to the supine position, tended to show larger changes in blood pressure. It's sort of like a global group level autoregulatory problems appears to lead to be visible in these subjects. And if we go back to these earlier things, perhaps all these things might come together and be related to outcome. Do we know this? No, not yet. This is sort of data mining and gathering data to get someone to fund you to look at a larger data set. I should also point out that this really appears to be related to injury, because we have also looked at patients with severe internal carotid artery stenosis, and we see the same type of correlation if we look at their affected hemisphere, the hemisphere where the carotid artery is more than 70% occluded. We also saw this, and not on the other hemisphere. So it appears to be a hemispheric problem, and it appears to be related to brain injury. We still have to find out what this also means for these subjects. For example, maybe we need to do an earlier surgery, even if it's riskier, or maybe we need to ask them to change their lifestyle even more drastically because they may have autoregulatory problems. Armed with this type of data, our colleagues in Hospital de la Santa Creu, São Paulo in Barcelona area said, can we try to use autoregulatory indices to see if they could help us pick patients better for clinical trials involving early mobilization therapies. Early mobilization has been shown to be positive for motor recovery, but the large AVERT trial result has shown that it may also have a deleterious effect on the neurological outcome. And if you look very carefully into this data, what you see is that they are suggesting basically that our patients are very heterogeneous, and that might be the problem. If we could personalize, there might be better findings. And they said maybe impaired autoregulation is the culprit. Since we don't know which patients are okay to stand up and be mobilized early, maybe we are doing them a harm. So they said, we're going to now, we are in the process of recruiting 200 patients, randomizing them according to their NIHSS score and other factors into two groups who are receiving the standard therapy or an intensive therapy, both physiotherapies. And we are doing a very sort of adventurous protocol with them by doing the first head of bed challenge, as I showed you, then going back to the original part, waiting for a longer period, and elevating the head of bed even higher, then asking them to sit and stand. For most of these patients, this would be the first time that they are sitting or even standing after their stroke onset. And this is decided based on the neurological and blood pressure status of the patients. Anyway, we try to do this also within the first 24 hours of the subjects. These subjects are different in the sense that our inclusion criteria is including subjects with lower NIHSS scores. You should keep that in mind. So another point is that we want to look at autoregulation indices, but we don't have all the invasive monitors that we had in the TBI data. We only have the non-invasive monitors. We do arterial blood pressure monitoring with a FinaPress, and we have TRS and DCS again. So then we started to look more and more into this problem, and then we realized that maybe we'll have to consider many other things here, correcting for untitled CO2 values, correcting for postures, because there were some data that said that different postures may cause different alterations in the position of the autoregulatory curve, which might have implications for us if we wanted to look at the whole period. So we wanted to look at each posture separately to assess CAR. And we wanted to look at what are the different ways we can look at it. So we noticed that people are looking at the typical plots, typical correlations, but they are also looking at the cerebrovascular reactivity, So MAP changes divided by CBF changes versus MAP. This should have a number like 1. And they are seeing that it has a big variability as well, and we can see what happens. And since we can't use our posture alterations, we had to look at the spontaneous fluctuations, which tend to be more significant in stroke patients than healthy subjects, but they are pretty low. Maybe we have to reanalyze this data like Wesley has suggested, and by putting on some additional thresholds. But anyway, this is what we did. We took windows in the data. We downsampled 40 Hertz data to 5 seconds. We took 150-second windows with 5-second shifts. And then we looked at both the CBFX values as before, but also the CBFX slopes, which is essentially the slope of the CBF versus MAP changes plot in each window. And we have also looked at the sort of this CVR changes versus MAP, which should have a value of 1, since CVR is derived from 1 if everything is autoregulated. So if we do this to the stroke patients, and if we again look at IPSI lesional and control-lesional sites and compare them to healthy controls, the gray shaded area is presumed healthy autoregulation, but we have no idea where it should really be. We see that the stroke patients have significantly higher values. Some have higher values, some don't. We are still looking through this data to see if the clinical variables correlate with this. Different colors here show different levels of NIH stroke scales and different types of strokes as well. But then, nevertheless, this is encouraging. The stroke patients show a larger variability, and they are different from the healthy in this parameter. If we look at CBFX slope, there's no significant difference, although the stroke patients still show a larger variability compared to the healthy subjects. If you look at the cerebral autoregulation slope, remind you this has to be around 1. We again see that stroke patients show a higher variability. They tend to have a lower variables, but the changes are not significant. But this is moving from this group level analysis that I have shown you before to individuals. And this is the values during the baseline. Now we could do it at different head of bed positions, sitting and standing, and look at all the details. So these are very rich data sets. We can do other things like looking at the presence and absence of low frequency oscillations. We will have to look at areas with significant MAP changes, as Wesley has suggested. We can plot data like here, one versus another, to try to look at it. We do see that the patients whose clinical status has deteriorated after the measurement tended to show a significantly higher CBFX, but we have to look more carefully to rule out any confounding effects from this. So now going back to a little bit to the nice methods that Jana has shown earlier. These methods are really interesting. Can we use ICP estimates obtained from non-invasive diffuse optical data to estimate cerebral autoregulation? So the method that Jana has shown is really interesting. She uses the blood pressure and optical data together, does pulse feature extraction, and uses machine learning to get to the point of interest that she has. In our case, we didn't use the blood pressure and we didn't extract any features. We had sort of blindly dumped a set of data into a machine learning algorithm, and we looked at the output. So our approach is as following. So we take data. We have a data set from adult traumatic brain injury patients, and we have a data set from children who are about 2 years old with an invasive ICP monitor throughout all night. We use these data sets separately, so we'll focus on the TBI. If you're interested in further, you can look at our publication. We use some parts of this pulsatile data as the training set, and we use some other parts of the test set. We use a recurrent neural network. The details are way beyond what I can explain in this short time. And then we have been obtaining good results, similar to the results Jana has found. This data actually has additional data compared to the paper. Now it has 12 TBI patients with 17 measurements, and you see very nice correlations on the left with the predicted ICP with invasive ICP. We can detect ICP wave, we can detect individual changes, and we have a bias, very small bias, 0.15 millimeter mercury, and an accuracy of about 3.9 millimeter mercury in this assessment. And as everyone else using this type of training data is saying, we need larger training sets. So we have actually started to acquire some from our colleagues at Emory University, from Erin Buckley's lab, from subarachnoid hemorrhage patients. They have non-continuous ICP readings, so we couldn't use them for training, but we have used them for testing using our training model. And we are also working with CHOP and PEN with Arjun Yod and Wes Baker, Andrew Kofke, Daniel Licht, and others to see if we can rely on them. And I'm not bold enough today to show you this data, but we are trying to use the learned model to apply to these stroke patients and such to look at kind. And in really quickly also show that some technological things that we are trying to do. So in this DCS blood flow measurements, we have these multi-center stuff happening, clinical trials. We are getting into medical device approvals combined with EEG and ultrasound. There's fast, high density, etc. But one also interesting area is trying to go small and low cost. So the question is, can we actually start to do things like what you see from the wearable NIRS devices using DCS-like techniques to measure blood flow without spending a lot of time showing how we do the technique? Let me say that we are working using something called speckle contrast optical spectroscopy, which is sort of an integrant measure of the DCS signal. And we were able to build the device that you see on the left. So we were able to develop our own long coherence laser, use a Raspberry Pi for a control and to send wireless signals, control custom-made single photon avalanche photodiode detector array containing 64 detectors, put it in a very small device with an FPGA implemented analysis algorithm and put it on subjects. And we are working on turning this into what you see on the right picture. Hopefully in 2 years we'll get there. And from this, we are doing measurements like you see below. People are now trying to be more mobile. We have wireless data, so people can walk around with the examiner, and we'll try to start to look at autoregulatory type indices from these subjects as well. So I would like to end here and check answered questions. So thank you.
Thanks a lot, Turgut, for an excellent talk. I know we are over. I don't know if Pat or Caroline have to make some announcements for next time, but at least I want you to answer at least one of the questions, which was, did vessel recanalization status have any impact in your results in the acute period? And do you have any experience with large vessel occlusion and low NIH stroke scale patients?
Right, that's a good question. So we are looking at that. We have sorted the data, especially in the early data sets that I have shown with the simple adenoid alteration. The recanalization status didn't pop out very clearly. But I should point out that many of the subjects these days are receiving the endotractomy, etc. So they are actually having recanalization. The stroke paper, the paper that was published in the journal Stroke by Favila et al., we describe a lot of details. In this new data set, in fact, these people do have lower NIHSS scores, scores like 4, 5, 6, instead of the 20s, scores in 20s that were in the earlier data set. So we have been measuring those, and those are the subjects where we are calculating the autoregulation indexes and such. Those are the ones who can be mobilized 24 hours into their stroke. So hopefully soon I will have data to show about the clinical correlations and such from those subjects.
Yeah, very interesting. So I'm sorry, Turgut, to only give you one question because I kind of need to move on. But I am sure if I speak for everyone in this session that please reach out to us. If any of the methods we're using sound appealing to you, reach out. If you have any questions about the optics, we're all more than happy to answer additional questions. Just shoot us an email. So thank you for the attention, and I'm going to hand it off to Pat.
Thank you very much, Jana, and thank you very much to all the speakers for a very, very interesting seminar. For upcoming seminars, unfortunately, we have some last-minute details, so it won't be possible to announce the next seminar, but expect to have a follow-up soon on the social media. So again, thank you very much to everybody.
Thank you.